Hi, welcome to Obesity Machine Learning competition, tackling metabolic diseases, and thank you for joining us. My name is Thiago Batista, and I'm a senior scientist at the Broad Institute, where as part of the Broad Diabetes Initiative, I work to apply functional genomics to understand the genetic risk of cardiometabolic diseases. Together with Julie Laffey, we've put together a three-part lecture series to introduce adipose biology, its role in metabolic diseases, and how we may use machine learning to better understand and treat these complex diseases. In recent years, our ability to perturb genes as a way to understand their function has dramatically expanded. Now, by bringing these approaches to disease-relevant cellular models in combination with complex, high-dimensional, and multimodal data, we have the unique opportunity to convert disease biology insights into therapeutic solutions. However, to interpret and derive translational insights from such complex data, we also require advances in machine learning methods. And that's where you will have the chance to bring your skills to really make a difference. So let's get started. First, I'll give an overview of the crash course. We'll have 3 lectures going from the biology of adipose tissue and its contribution to metabolic diseases. Then we'll follow to talk about the technologies to perturb adipocyte biology. And we'll talk about the data that we'll assemble for this competition and the 3 crunches that we'll be working on during the challenge. So let's start with the biology of adipose tissue and its role in metabolic disease. In this lecture, we'll cover 3 key topics. What is adipose tissue and metabolic diseases, and how is adipose tissue formed? So what is adipose tissue? Adipose tissue, or most commonly known as fat tissue, is found throughout the body, either right under the skin, or what we call subcutaneous adipose tissue, or in close proximity to the internal organs. That's what we call visceral adipose tissue. The main cell type of adipose tissue are the adipocytes, which can be subdivided into white adipocytes, brown adipocytes, and beige adipocytes, which will share functions with both white and brown adipocytes. And adipose tissue serves many functions to support our body. It can provide thermal insulation, so we can maintain our body temperature. It provides mechanical protection and intra-organ cushioning, and also it provides a safe harbor for lipid storage and handling, which is linked to the many metabolic functions of adipose tissue. Adipose tissue, particularly white adipose tissue, it's a major site for lipid storage and release when necessary. After a meal, our food will be converted into glucose, amino acids, and fatty acids, which are a kind of lipids, and reach the adipocytes through the bloodstream, which then will be converted into lipids and stored under structures that we know as lipid droplets. Now, under conditions of prolonged starvation, cold exposure, or extensive exercise, our body will send signals to the adipose tissue so these stored lipids can be released into the bloodstream and reach distal organs to be utilized as energy sources. And in between cycling this storage and release functions, adipose tissue plays a critical role in the regulation of energy homeostasis and maintaining blood glucose levels. In most cell types, including white adipocytes, nutrients are processed in the mitochondria by the TCA cycle, which will result in energy production in form of ATP. Now, in brown and beige adipocytes, there's the expression of a special protein known as uncoupling protein 1, or UCP1, which can bypass this energy-producing step and generate heat instead. We call this energy-dissipating process thermogenesis, and it's currently under active investigation by researchers as a tool to promote energy expenditure to combat metabolic diseases. In addition to energy storage and thermogenesis, adipose tissue also contributes to metabolism through communication with other parts of the body. This involves the release of bioactive molecules, which will include peptides and hormones, metabolites, inflammatory cytokines, and others, which will enter the bloodstream, reaching distal organs such as the brain, muscle, liver, and others, what will regulate functions including appetite, energy expenditure, and others. In addition to adipocytes, adipose tissue also contains many other cell types that will support its function. Recent progress in single-cell transcriptomics, which is a method that enables us to measure gene expression patterns in individual cells, has dramatically expanded our understanding of the cell types that compose adipose tissue. So here's a general workflow where we can go from sampling tissues from donors. We can either process or freeze those for later experiments, and then we can go under single nucleus or single cell sequencing workflows. With this data, we can map not only the adipocytes, but other many cell types that will include endothelial and other vascular cell types, many classes of immune cells, and also adipocyte progenitors.